Welcome to JMBE live from ASM Micro Meeting in Atlanta. We're really happy to have you here. And today we're going to be talking about CURES, course-based undergraduate research. This is a really wonderful opportunity for ensuring that every one of our students who graduates with a degree in science understands how science is done. As you all know, the number of opportunities for students to do research with faculty members is always somewhat limited. And that means that some students will go through their entire education and not have that experience. The three people we have on this panel today have been doing their part to change that at their institutions so that all students can get this experience, know how science is done, and that is really critical for this distinguishing evidence-based science from pseudoscience. I'm going to have each one of the panelists here introduce himself. Would you begin, Katrina?
Sure, of course. Hi, everyone. Dr. Katrina Twing. I'm an assistant professor of microbiology at Weber State University in Ogden, Utah. I am a microbial ecologist at heart, but I'm in a micro department and teach a whole variety of different classes there.
Hi, everyone. I'm Samantha Parks. I'm a principal lecturer at Georgia State University, so just down the street. I'm in the biology department, and I'm also a microbial ecologist.
Hi, everyone. Good morning. My name is Jeff Olimpo. I'm an associate professor in the Department of Biological Sciences at the University of Texas at El Paso, and I also direct our institute's Teaching and Learning Center. By content training, I am a developmental biologist and a virologist most recently. But I consider myself a discipline-based education researcher, and that's the work that I do at my institution currently.
So each one of these people has a very different level of experience, different opportunities. So we'll begin. Katrina, could you talk about what you've done to develop your cure? And it'd be really nice in the process of saying, tell the audience what a cure is. How do we define a cure?
Sure, absolutely. So I currently have a semester-long CURE, course-based research experience for my microbial ecology students. That's a required class for all of our majors, so everyone passes through it eventually. And it has a lab component, so they're taking 3 hours of lab every week. And I decided that I wanted them to be active researchers, starting out with, learning the scientific method from asking a question all the way through interpreting their own data at the end of it. It is exponentially more work than a traditional lab course. A lot of times in a traditional lab course, you have a lab manual. Even if the students don't know the outcome of their experiment in that given day, the instructor does. And so it makes everything really straightforward from assessment as well as just planning and prep. As a lot of us know, when you're doing active research, you may not know what methods you need in that moment or where the data are going to take you. And learning that alongside some students that you're also teaching the fundamentals of the methods can be challenging. When I started my position, I knew that I wanted to do a semester-long CURE because I had seen in just a small tidbit what a short-term one of maybe 3 weeks had really done to give the students ownership over their science. I'd seen the cookbook method where they kind of came in and did, what was in front of them and left. And then a few weeks of doing a mini CURE just really light up with that ownership and feel like they were, but feel like scientists for the first time. So I wanted that in a full semester. And the one that I implemented when I first started was the PAIR project, the Prevalence of Antibiotic Resistance in the Environment, out of Tufts University, already established. But it was a lot of work to do that, and then my individual research, and they were diverging, almost doubling my workload. So I've recently restructured, figured out who I want to be as a scientist and an educator, and developed a whole new project, streamlined, where that data is feeding in. I'm sorry.
So that was really a great perspective of the amount of work required. I guess I would like to just point out that whenever you teach, if you're passionate about teaching, it takes a lot of energy and effort. And it's so nice to know you're having the real impact at the end of the day, even if it takes more work. So
Absolutely.
right. So Samantha, you have had this experience for a while and have really encouraged your university to embrace cures. Could you talk about that experience?
Absolutely. So we are a very large department, over 2,000 students, and we don't have enough labs for them to all get research experience. And so it becomes very difficult. It was also very difficult to convince the university for individual faculty to take on these small labs of 20-ish students, and we needed to show the value. And so we started small. There were maybe 3 of us, where we'd each take about 15 to 25 students, have them walk through hypothesis development, making up their own methods, starting to find their own data, and then present it. Those small cohorts of labs have increased exponentially. We even have a microbial ecology consortia, so several different labs that build on each other. Students can take several labs over the course of their time as undergrads. We even have students now who have been undergrads who have become TAs and taking this on into their PhDs.
So scaling is one of the real big issues, right? And Jeff, you're doing a lot to try to figure out how can we scale this so that even at very large universities with many students, we can reach them all. Would you talk about that?
Yeah, absolutely. So I would say that we started much in the same way as Sam did, offering pilot sections of a particular cure. When I first started out, I was engaged in and teaching a marine ecophysiology cure. And one of the things that we found there that helped us in scaling is to kind of front load a lot of the logistical elements. So if we needed equipment or supplies, etc., we were very intentional in ensuring that we purchased those early on, and then as future semesters approached, we could sustain the cure essentially by making use of student lab fees. So all kinds of consumables and things that were in that space were then paid for, if you will, out of the student fees that were paid for the laboratory curriculum. I think the other thing that's really critical in scaling up, which kind of echoes the point that Sam made, is this notion of engaging, in our case, with teaching assistants or other instructors for the course. So in a multi-section experience, at least in my own institution, all of our laboratory curricula are taught by TAs. These are graduate students in our case. And so engaging them in professional development was really critical around things like mentoring, what makes cures unique, helping them develop certain research skills if they were teaching in a class that was outside of their comfort zone in terms of research familiarity, and providing the context for them to be able to engage with us, me as an outsider, right, as an education researcher in that space, but also instructors of records and other things when they had specific questions or concerns that were not simply just content-oriented questions or concerns. So I would say in terms of scaling up, start small. That would be my advice. Start with a few pilot sections, work out the bugs or kinks, and then really ensure that you have the appropriate infrastructure in terms of resources, personnel and otherwise, that's going to support the scalability of that experience. And I would say lastly to the topic, right? I think the other thing that we found was really critical is ensuring that the topic was broad enough that there was lots of opportunity for discovery, right? Novel work being done, but also not such a steep learning curve that the students and consequently the instructors in those spaces, GTAs or otherwise, were going to come up against that as a really key barrier that they had to challenge.
So let me follow up on that, Jeff.
Sure.
And so one of the criteria for being a course-based undergraduate research experience is that you're doing real science. There's discovery. The students are finding out things nobody knew before, right? It's not something where you knew and you're making them think that they're discovering something new. They really are discovering something new. 2, that they can iterate. When something doesn't work, they go back and do it again and make it work. They can design the experiments to be to address a certain point, right? And And they learn how to deal with messy data, right? It's not just like a canned lab where you expect the data to look like this, and that's probably the way it's going to look, right? So these are real research experiences. And sometimes the students can present them on posters or publish, and I think that's an experience all of you have had, right? So, so it's when you talk scaling, this issue of iteration is one of the kinds of things that gets to be very important in terms of thinking of scaling. It also influences the investment of the university in these programs. And it would be really interesting to have you say a little more about how you got buy-in from the leadership of your university. And why don't we start with you, Jeff, and we'll come back this way, because you're all at very different institutions.
Sure. So I actually paid them all off to get buy-in for things. No, just kidding. I was very fortunate when I stepped into the picture at UTIP that we had a growing culture, essentially, of interest in course-based undergraduate research experiences. We are in a similar position where it's just not feasible at all for all students to be involved in faculty-mentored research or apprenticeship-style research. Though we have a large number of faculty, we also consequently have a large number of biology majors and serve a huge number of majors across different disciplines. So in terms of buy-in, I think our biggest hurdle, first off, was the why question. Why would I invest a lot of additional time and energy to teach this course when I could simply teach the traditional laboratory equivalent and get the same, we call them TLCs, teaching and learning credits, right? Or FTEs for teaching and learning. And we leveraged, I think, the point, Katrina, that you made, which is to say at our institution, we model our cures off of UT Austin's FRI model, by which there are research streams. So an individual faculty member leads a research stream based on their own research interests. And so we really leverage that aspect to say, well, you know, this not only has benefits for students in your course, for the TAs you're working with, it can contribute to your own research productivity. There's opportunities to highlight mentorship in a context outside of your laboratory space, et cetera, et cetera. And oh, by the way, there's a support system in place to help you out, right? So that was our other primary challenge, was individuals who had no idea what a cure even stood for, let alone what the components that needed to be baked into a cure were. And so understanding that there was a whole team of individuals behind the scenes and also on the scene, right, to help them, I think was really key in pushing them kind of into that space. And then knowing that once they started in developing and implementing a cure, that the support infrastructure was not going to disappear was also really critical. So those would be my big things, having the background and on-the-scene support infrastructure, and also leveraging buy-in in terms of advancing their own professional growth as individuals at the institution.
So to echo a little bit, we did visit UT Austin and try to kind of see how they had incorporated these labs. We struggled with buy-in. So we started by kind of going to labs that were closing down at the lab at the university and gathering some of our own equipment that way, kind of carving out a little niche. And our big selling point was about publishing, because part of working with CURES, since it is authentic research, it was a way for our students to present their data, but also for faculty to publish their ideas, for faculty to start doing some of this educational research and really expand. So from that initial cohort, as we've expanded out, we've had several faculty start to publish. We've even had some graduate students publish in Tips and Tools for JMBE with their ideas for how they have started to present this idea of authentic research to students. So it was buy-in from the university, but it was also getting that buy-in from students that really became important, showing students the importance of doing the research and also the possibility for presenting at Abrahams, at ASMQ, and presenting their work through, kind of local conferences, even at Georgia State, and then starting to publish their ideas.
Before you go on, Sam,
Yeah.
Another thing that, since you've brought this up, it'd be nice to know about evaluation of student learning. Is this more effective? Does it have the same effectiveness as traditional labs?
So We've done some research investigating this, and sidebar, we all talked a little bit before this. So, Katrina, one of the first things that students talked to me about, one of the initial cohorts of Georgia State students is, they said they wanted to be assessed. They wanted to tell me their opinions. And the biggest thing was that they felt that they were part of the scientific community. So as a student in a didactic classroom, you kind of sit back and you hear information and you take a test and you're done. The students, we forced them to get up. Before they went to a conference, there was a hub in the middle of Georgia State's library, and we said, you have to give a talk. Get up there and do it. And they were terrified. But when they stepped down from that platform, they were scientists. They had just shared information that nobody else had ever known. They shared pictures of bacteria that they isolated from Winogradsky columns that they tested with antimicrobial compounds. And it was a moment of pride in our students that I had not seen. I've been teaching on and off for 20 plus years, and that moment still gives me chills. So when you're assessing, you can assess lab skills, you can assess presentation skills and writing skills and all of that, but there's something else to assess, and that's a sense of belongingness. That's a sense of being part of a scientific community that I don't think you can get elsewhere.
Wonderful. Absolutely.
So, Katrina, how about you? I mean, so you came in as an assistant professor. So for you to get buy-in, they'd been around for a little while. Might have been easier for them than you.
I came in as an assistant professor during the pandemic at a primarily undergraduate institution where there is a lot of support. Let's talk about different levels of support. There's a lot of enthusiasm for high-impact educational practices, but there is limited monetary support. And as you were mentioning, right, it's very expensive to get started up. I had my startup funds limited at, a non-research institution, and some of that was useful. I have been financially supported in developing my CURES by the university. We do have kind of a panel. I have a attribute that my course is a CURE that goes through kind of an application process, and that allows that to be on the students' transcripts, which is really important, but also for me to be able to apply for funding. With that being said, it's a very small pot of funding that as more people across campus are beginning to use these really essential tools, that pot of funding hasn't changed at all. And so while I have been supported, there's questions of whether or not I'll be able to be continued to be supported to do this. And grad students, oh man. I'm, again, doing all of this with grad students and limited funds. So.
All right, let's shift to something a little happier. So Samantha said that one of the real benefits is this sense of being a scientist, the sense of belonging. You had mentioned that you see another benefit to this. And that really has to do with inclusion.
Absolutely.
Would you say something about that?
Yeah, absolutely. So one thing that I think we've kind of highlighted, but a CURE experience allows every single person in that classroom to be a scientist, and maybe more importantly, to see themselves as a scientist. We're an emerging Hispanic serving institution, and our texts are trying to catch up, but we're not seeing representation very much in examples, in textbooks, even sometimes in the publications that we choose to share with the class. And so one thing that I found really valuable in using a CURE is that the students, regardless of their background or what they look like or what they've seen modeled, can see themselves as a scientist. And one young, I had one student, she's first gen, Latina, part of the LGBTQ plus community, who ended up winning our Student Researcher of the Year award for the university. She commented on the fact that she didn't see herself as a scientist didn't think she could do it until I saw her as a scientist and showed her that she could do it. And still brings tears to my eyes to think about that. But that is why the work, the money that goes into it is so worth it, and why we need buy-in beyond just individual instructors doing it. It needs to be institutionalized and funded.
So, Jeff, we've heard Sam's perspective and Katrina's perspective about why this CURE experience is so valuable. Can you add something else to that that would argue everyone needs to do this?
Yeah, so I would say I wanted to comment first on the assessment piece. I think you need to ask. I tend to be heavy on more qualitative leaning data because you're going to get an understanding of belongingness or sense of identity or whatnot. You can't do that with a Likert item scale, really. I mean, you can, but I would encourage you to actually pursue other forms of assessment that are more open-ended. For instance, prompts that students fill out, collecting artifacts, things they generate in class, and understanding those pieces as well.
Jeff, can I interrupt you?
Yeah, of course.
Do you, as part of your mentorship, do you have templates for that people starting out can kind of use and develop for their classes?
Yeah, absolutely. There are a number of really great tools that are already published tools that will aid you in looking at these things if you're interested in adopting more of a quantitative approach, if you're more qualitative leaning. Honestly, you can just email me, frankly, right? We have a whole repertoire that we use to analyze things that are more affective or psychosocial in nature.
I just want to warn you, JMBE Live goes onto YouTube. So many people will be looking at this for a long time. Your email box might get really full.
That's okay. Send away. I'm okay with that. I'm happy to share out, I think, because I say that very sincerely because I think the more we engage in that type of work, the more representative understanding we have to your question, right? What is the true impact and value? And I think we're there, right? I mean, it's been a decade since the acronym CURE kind of came into existence and started labeling courses. I would say for me, the biggest win is engaging with individuals who are non-science majors in my courses. So I think about the comment about science identity, and they're often like, "Well, I'm not a scientist. I'm a business major, right?" And so creating a space for those individuals to engage in ways that they're comfortable in engaging in doing scientific work, with no assumption about prior experience, knowledge, ability, etc. And having them walk out the door, right? I mean, I'm thinking about identity in a different sense here in terms of major, but having them walk out the door and feel like they made critical contributions, that they did actual science and didn't just pour two colored test tubes together into one, right? That kind of cookie cutter or cookbook model approach, I think for me is the most impactful. And I will say the other thing that's really impactful is we do not have the capacity at UT El Paso to offer cures in all of our we just have too many sections, right? Quite frankly, to be able to use the FRI style model across all of those sections. But it's the word of mouth that happens. So students take part in a cure and then it snowballs, right? It spreads and they tell other students who then tell other students, et cetera, et cetera. And by virtue of that, we create this kind of community of individuals who are engaged in research at all levels and are also mentoring their peers, right? Or showcasing opportunities for research for their peers. At UT El Paso, that's really important. We're kind of a unique campus. We're 85% Hispanic. I mean, that's kind of monolithic, right? But Latina, or Latinx. We're a commuter campus by and large. We're right on the U.S.-Mexico border. So we have a lot of very interesting demographic kind of attributes or features of our campus. And so we get a large number of students who are not even aware that research opportunities exist, right? And so that snowball effect and peer mentoring effect has been really powerful, I think, in immersing students in the research and scholarship culture on our campus.
So I just want to emphasize for the audience that your point about assessment is really key, because many of us who were not trained in discipline-based education research, we would always experiment and try new things with our classes, but in the end, it was a gut feeling about whether it was working better. And the change is really to if you're going to do an experiment, to make sure you assess that experiment, and if it is working well, then you can share it with other people and improve education around the world. And I think that that's one of these examples that it goes beyond the teaching in the classroom, which is very high impact and emotional, and allows other people to have that experience as well. And so I think this kind of discipline-based education research and what it takes, as you mentioned, Jeff, sometimes you need to go to an expert to get their perspective to be able to move it forward. And I'll just remind the audience that JMBE, the Journal of Microbiology and Biology Education, published by the ASM, is a forum for a lot of this. And Katrina, have you published in JMBE?
Not yet, but I do plan on
Not
it
yet.
very soon.
Not yet. But both of our other speakers regularly publish in JMBE, and so you can look them up and actually see some recent publications tied to these things. We've talked about this kind of high level. You mentioned what you're doing in your labs, but I don't think the two of you really went into any detail about what students are doing in this lab. It'd be good to give people an idea about what it involves.
So my background was in biodegradation of nitroaromatic compounds. And when I started at Georgia State, the students looked at me with this blank stare, like, what? And so I said, what are you interested in? And one of my favorite comments from a student was, well, every time I'm sick, my grandmother makes me drink yellow milk and I want to know if it works or not. Okay, so you're talking turmeric? Yeah. So this started a snowball where we now have well over 100 isolates of microbes growing on different non-traditional antimicrobial compounds, where we have isolated them using Winogradsky columns, supplemented with turmeric, cinnamon, tea tree oil, aloe, you name it. And now we've advanced to where we're starting to figure out how these microbes are growing on these compounds and starting to identify the metabolic pathways that they are using to break them down.
Jeff?
Yeah, for sure. Thank you. So I teach a full year health disparities cure intentionally because we were trying to bring in a large number of different types of majors. And so the students in the cure that I facilitate actually have quite extensive agency in determining what types of projects they conduct. So we've had students examining things like air quality in El Paso, which is particularly poor currently. I'm not looking forward to going back home, right? Particularly poor. We've had individuals who have an interest, for instance, in food deserts and trying to understand access. And the cool part about that is that it combines both kind of more traditional wet bench techniques, and then others who are more survey, right, oriented because they're looking at perspectives or they're looking at prevalence of certain things in the region. So the research-oriented component occurs in the fall, and then in the spring, we implement kind of a civic/community engagement component where students take their research findings and translate it into some kind of outreach piece. So for example, our group that was interested in food deserts started exploring opportunities to build up the food pantry on campus, to create a community garden on campus, to do things like that. So they can understand regionally, it's very place-based obviously, but regionally what the research translation component actually does, right? Rather than, I mean, there's importance to publications and presentations and generating new intellectual and theoretical capital, but a lot of our students are in the community. They've always been in that community, and so giving back, right, and supporting the community is really near and dear to their heart. So providing a space for them to do that, I feel at least, is really important.
Nice. Real world impact.
Exactly.
That's great. Could you just give us the 30-second version of the very first cure you started?
Oh my goodness. I've been involved in a number of them. The first one I remember being a part of was this marine ecophysiology cure. Right. So I worked with copepods, which I think are little sea critters, and that's a really terrible, I would get punished for describing them that way. But they provide a really great model system to do all kinds of studies. So students were doing toxicology work. They were trying to understand other factors that impacted like hatching, right, or reproductive health of these organisms. They were changing salinity conditions in an organism that's really very understudied. And so it was a great way to get people roped in early and doing research that again didn't present a really huge learning curve for students in this case that were in an introductory biology space.
Great, thanks.
Sure.
I wanted that just so that people could see the variety of different things that are working in CURES and the way to really engage students and have these impacts. Our time is about up, but I want to say thank you very much to all of the panelists here. And thanks to all of you who are here in person. And we look forward to, if you are an educator and you are doing research, science-based education research, we'd love to have you contact us and publish in JMBE. That's the way your impact in your classroom can have a much broader impact across the entire higher education community. Thank you so much. If you have a friend who wants to look at this, JMBE appears on YouTube about a week or two after we've recorded a session, so check it out. Thanks very much, folks.